All right, guys, welcome back. Hopefully I haven't scared you off after our 20-minute lesson yesterday on the first part of cell organelles. Today, we are going to be referring back to our anchor phenomenon about endosymbiotic theory. We'll talk about what's involved with that and how we got there. Then we're also going to talk about some of these organelles that kind of lead us to believe this theory, which would be the mitochondria and the chloroplast. And then we will talk about some of the support structures in a cell because they don't really go anywhere else. So we'll just talk about them today. And depending on how long this video goes, I'm going to try to fit in a discussion of the project. If not, I'll make a separate video for that. But let's get started. Okay, so endosymbiont theory. That's a big, nasty science word, okay? Endo means inside, sym means together, and bio means life. So they are living inside together, literally. So what this means is that what we're trying to do is explain why mitochondria and chloroplasts look suspiciously like a prokaryote, like a bacteria. I don't know if you realize that, but those particular organelles are way different than anything else in the cell. So let's talk about some of those differences. So this theory says that a long, long time ago, a eukaryotic cell engulfed or ate a prokaryotic cell. So it surrounded it, but it didn't kill it. It ended up surviving, like the bacteria survived inside the eukaryotic cell as an endosymbiont, so a cell that lives inside another cell. We can still see this today. We used the word protist in the last lesson, but a eukaryotic single-cell organism, and it can have algae cells that are living inside of it. And they've run tests, and those ones that have the algae, they survive better than ones that don't. They're resistant to starving and all those kind of things. So we can still observe this today. It's just the ones in animal cells, plant cells, they've been there a bit longer. Okay, so eventually they became one organism after they lived like that for long enough. So some of these pieces of evidence, I'm going to go ahead and put them on the screen and then we'll talk about them. So both of these organelles have a double membrane, and when you look at them under a microscope, like an electron microscope, you can see there's two separate outer layers. Sort of like what would happen if a bacteria was surrounded, like when endocytosis happens, like something gets eaten by the cell membrane folds around it. So it leaves like a layer of cell membrane around whatever it engulfed. So we're still seeing remnants of that membrane around these organelles. They also have their own ribosomes. They have circular DNA called plasmids. We see these in bacteria usually, but a chloroplast and a mitochondria both have these to this day. And they're also capable of functioning on their own. They do their own thing, they divide, they do all of that on their own. Just really kind of cool. So the DNA of mitochondria is actually kind of important when you're wanting to test lineage, like 23andMe. A lot of times they look at your mitochondrial DNA because that goes through the mother's line and you can trace back and back and back that way. All right, so the first one, mitochondria. If you write it's the powerhouse of the cell on your test and nothing else, I'll laugh at you and then give you no points for that question. So in AP, we want to say smart things like it's the site of cellular respiration. We're going to learn in cell energetics, which should be unit 3, that's where we break down sugar into something called ATP, which is our universal energy molecule. So the structure of this double membrane on the mitochondria, the outer one's smooth, which is why we're always drawing it as this funky bean shape. But the inner membrane is all folded, and we call these folds cristae. So this divides the mitochondria into two internal compartments, and it also increases the surface area. It's kind of like how your brain has folds in it. These folds are able to increase surface area. So the intermembrane is the space between the inner and outer membrane. So that would be the lighter orange on this picture here. Okay, so inter means between, so it's between the two membranes. And then there's the matrix, which is enclosed by this inner membrane. This is where the Krebs cycle happens. So the matrix is kind of like the liquid-filled inner part of our mitochondria bean, so to speak. So Krebs cycle, respiration. Get that in your head. You're gonna need it. So also in the matrix, there are enzymes that catalyze, or remember that means reduces the activation energy for cellular respiration, and it produces ATP. So actually, cells without mitochondria cannot do cellular respiration. They do something else. So also in this matrix, inside the inner membrane, we have the DNA and we have its own ribosomes, which is kind of cool. A mitochondria doesn't need to wait for the nucleus to make proteins that it needs. It does it on its own. It's a self-saving princess. So the number of mitochondria in a cell correlates with its metabolic activity. Basically, if you need more energy, you have more mitochondria. So cells that move or contract, so your muscle cells, your intestine cells are moving all the time. They might have more mitochondria than, say, a fat cell, because fat cells don't have to go anywhere, but your muscle cells got to do all kinds of things. So here is a scanning electron microscope, well, not scanning, but it's an electron microscope image of an actual mitochondria. So we can see along the outermost edge, there are actually two layers of membrane. There's an outer one and an inner one. We can see the cristae, like how the inner membrane folds in on itself like that, and then the matrix is the area in the middle where all the fun happens. So we didn't really talk about it right now because that's, cellular energetics, but there are things for respiration and energy production that happen in the folds, but we'll leave that for another day. All right, chloroplasts. So these are found in photosynthetic organisms. It's the pretty green bean shape that makes photosynthesis happen. So there's a green pigment called chlorophyll inside. So in this double membrane, there's something called thylakoids. Okay, so we see these stacks of green discs, right? These green pancakes. Each pancake is a thylakoid. Okay, so one pancake is a thylakoid. The whole stack of pancakes is called a granum, okay, or a grana. So this is important because this is what's capturing sunlight in here. Think about it like if someone is doing a high jump or a fall, they have like layers of cushions to absorb that energy and catch them. That's exactly what's happening with the granum. It has stacks of thylakoids to catch like basically the photons of light and turn that energy into something else. Go up a second. Okay. So stroma is fluid found around the thylakoids. So it's the matrix of the chloroplast is stroma. I think like stroganoff, like the liquid part that it's got inside of it. So this is where the Calvin cycle happens. So the Krebs cycle was respiration. The Calvin cycle is photosynthesis. This is where we're taking the words like an intermediary molecule. So what I made right after I captured the sunlight goes into the Calvin cycle and gets built up into a sugar. So the stroma also contains the DNA and the ribosomes and the enzymes. Enzymes do everything, just get used to that, literally everything. So we have a couple photographs, not computer images, these are actual photographs of a chloroplast. So the top image, what has happened is they have used special stains to attach to the different parts, and then you can put a special light on them and it makes them fluoresce or glow. So they used a special stain that made the chloroplast glow red. And so that's what we're seeing in that image, or all the chloroplasts in that particular algae cell. And the bottom black and white image, this is a single chloroplast up close. So we can see the stacks of thylakoids right there, the granum, and we can see the stroma, which is like the in-between bit, okay? All right, cytoskeleton. So the cytoskeleton parts, it doesn't really go with the energy stuff, but we do need to know about the structure of a cell and the things that help it keep its shape. So the cytoskeleton is a network of fibers through the cytoplasm. So they give structural support, so basically helping a cell keep its shape, and then mechanical support, which we're going to see in biology. This doesn't refer to machines. Mechanical and motor, all of those ones refer to movement, okay? Because there's not really an engine in a cell. So this movement support, I can either prevent organelles from moving or I can help them to move from one part of the cell to another, because like vesicles, those need to move so I can get things in and out. Sometimes I might want to move other stuff too. So movement can occur when the cytoskeleton interacts with motor proteins, so kind of like a zip line. Okay, so there's three main types of fibers we're going to talk about in this section. We're going to talk about microtubules. We know micro means small, and a tube is a tube, okay? Microfilaments, it's a small filament, like a small string. So tubules are hollow and filaments aren't. And then intermediate filaments, they kind of, they're somewhere in between. So microtubules are rod-like structures made of something called tubulin, because scientists are not very creative. They grow from the centrosome. So they assist in microtubule assembly. So centrosome, this is similar, I'm pretty sure it's just the same thing as a centriole, if you've heard that before, because we use these to reel things in. So like when separation of chromosomes, that's a centriole. So think of it like if a centriole is like a reel, the microtubules are your fishing line, and you can use them to drag things in or move them around. So these microtubules can be tracks for moving things. They also pull apart chromosomes during cell division. So if you recall from biology or life science back in middle school, when you do cell division, there's like the little X's, and then the lines get drawn and they make the X's break apart. Well, those little lines are microtubules. And they help in cell motility. Remember, if I see motor or anything like that, it's movement, cell movement. So cilia and flagella are made of microtubules. So here is a zoomed in picture of a flagellum, which is a whip-like tail in a cell. Sperm cells, some free-living ones, some bacteria have these flagella. And we can see like the inner core, that's a microtubule. Okay, so microfilaments are made from the protein actin. They help maintain the cell shape and by bearing tension. The way I think of this is if you've ever been in a big tent, sometimes they have to use ropes to help hold up the roof in a really big tent. So they are bearing the tension or the weight of the canopy, and so they help it keep its shape that way. It's kind of the same deal. They assist in muscle contraction and motility. So the actin and the microfilaments works with another protein called myosin to cause a muscle contraction. They kind of move over each other when muscles contract and relax. It also helps in the division of animal cells. So there's something called the cleavage furrow. It's okay if you're giggling, I can't hear you. So the way that we get animal cells to pinch off, like they started out as one and then they, broke apart, that happens with microfilaments. Okay, intermediate filaments, they have varying subunits and they're permanent structural elements of a cell. So they're not assembled and broken down at the rate of microtubules and microfilaments. I can build those up and break them down quickly depending on what I'm trying to do, but intermediate filaments stick around. So they maintain cell shape, they anchor the nucleus and other organelles because we don't want those going anywhere, and they form something called the nuclear lamina or the nuclear envelope. So it's like the inner part of the nuclear envelope. Okay, so looking at time, I'm going to talk real quick about your project. Okay, so what you're going to be doing is we are going to elect a most important organelle of the cell. It's been the nucleus for a long time, but the nucleus has some competition. So this project is going to be happening in stages. I don't expect you to do this entire thing at one time. That's why there's going to be different due dates for different parts, and it's on the calendar. So this should all culminate in us actually having an election the day before election day. And you're going to have all your campaign materials you've made, and we're going to let your class and Biology 1 vote on the most important organelle, because they'll be done with their cells unit 2. Okay, so just you can read this, but basically you're going to be working in a group of no more than 3. And you're going to run a campaign to get one of these organelles elected. So what's going to happen is on the day of topic selection, right, the day when this assignment first goes live, okay, starting at 10 o'clock, you can send me an email with what team you want to be on, and it will be first come, first serve with the timestamps on your email after 10 o'clock. If you are getting into my assignment at 8 and trying to cheat on that, or get ahead while other people aren't paying attention, I won't count it. Like, I'll just delete your email and try again after 10 o'clock, okay? You will be expected to work with your group, arrange a Google Chat or a Zoom call or something. I'm going to keep a document on there, like you can keep referring back to see who has signed up for what. But keep in mind, if you don't sign up by the end of the class, like the end of class, I'm going to start just assigning people because you'll need to get started right away. All right, so the first thing that you're going to turn in, well, rewinding, the first thing that you should do after you've gotten on your team, okay, is I want you, each person, to research all the organelles. Keep your sources, because you're going to have to submit your sources to me. Now, this exact write-up may change from this video a little bit, because you have to realize I'm speaking to you from 2 weeks ago. So things might have changed a little bit since then, but in general, what you're going to work on first is research, which is probably today. And then tomorrow you're going to be making a campaign poster, and that's the first thing I want due. So let me show you examples of campaign posters, if I can get the little bar to go away from Zoom. Okay, so these are real life campaign posters. You notice on here, there's not some big block of text really. You've got a picture, a few words, because when someone's walking by or driving by, they don't have time to read an essay on why they should vote for you. You need to have a slogan, a picture, something to make them vote for you right away. Okay, so this poster is what's going to be due. Work with your group. Okay, I'm going to be asking at the end of the project for people to evaluate your group effort. And if you didn't help, you can't expect the same grade as the rest of your group mates. I'm willing to allow maybe working alone, but keep in mind, this is an awful lot of work to do by yourself. You're going to want to cooperate and work together and divide the workload. So this is the first deliverable, is a campaign poster. Okay, the second deliverable thing that you're going to have is a smear campaign pamphlet. So like when you see the political ads and all they do is trash talk their opponent, that's what I want you to do for organelles. And every member of your group has to submit a different one. So I'll talk about that more in a second. Your third thing you have to make is a campaign video uploaded to YouTube. I know some classes have you uploading, accept TikToks, but the problem is I want people on election day to be able to watch your video and decide who to vote for, okay? So it needs to be YouTube so people can watch it on the school computers. So that's gonna be due on the 29th, so the end of October, right before our election. So your poster needs to be in color. It can be on paper or digital. Okay, and if you want to do more than one design, that's fine. I know this video is going long, I'm sorry. So provide information about your candidate's strong points, but don't put a block of text on your poster. This is due at the end of class on Wednesday. Okay, you'll have all during class to work on it. So I'm going to probably, in this video here, I'll talk more about what's due when the other due dates approach. I'm going to make all of these parts open in case you work ahead, but don't make me regret it, okay? I'm going to be looking that you all do this right, work together and get it done. This should be a fun activity. Please let me know if you have any questions, and I'll see you all in the next video.